Hi, we're going to talk about the alveolar gas exchange today. And at the end of today's lecture, there are two things I'd like you to feel like you've accomplished. The first is, I want you to be able to describe how a molecule of oxygen travels from the atmosphere to a red blood cell, and vice versa, how a molecule of carbon dioxide travels from the red blood cell back to the atmosphere. We're going to do that by focusing on a couple learning objectives. The first way we're going to break up learning objectives is through anatomy and histology. You're going to be able to describe the difference between total pulmonary ventilation and alveolar ventilation. You're going to be able to diagram the components of the alveolar capillary unit and describe how these components can be affected in gas exchange. And finally, explain the role of surfactant and describe normal alveolar refreshment. The other part of the learning objectives will be physiology based. By the end of today, you'll be able to write the alveolar gas equation and explain its use, define the respiratory quotient, define the normal AA gradient for the partial pressure of oxygen, and as a thinking exercise before we go further, you're going to be able to compare and contrast how VQ mismatch and hypoventilation affect the AA gradient. Let's start with describing the difference between total pulmonary ventilation and alveolar ventilation. Another word for total pulmonary ventilation is minute ventilation. Minute ventilation is made up of two components: respiratory rate multiplied by tidal volume. Now pause here. Imagine that your tidal volume is 500 milliliters, which actually is the approximate tidal volume of any adult. Count your respiratory rate for a minute, and then multiply the two together, and then you come up with your minute ventilation. I'd encourage you to pause the tape and actually do this. Assuming you're not too anxious, maybe you breathed 15 times in the past minute. So 15 times 500 is 7.5 liters, which is probably an average minute ventilation for any adult human. Now the trick is that tidal volume actually is composed of two different parts. There's the dead space volume, which comes from the conducting zones of the airways where no oxygen or carbon dioxide transfer occurs, but those things are transported back and forth from the atmosphere. And then there's the alveolar volume, where actually oxygen carbon dioxide exchange takes place. One interesting thing to note is that dead space volume is normal in all of us. But when you become sick, you can have an increase in dead space volume, which would be pathologic dead space, and therefore reducing the amount of actual alveolar volume. What we really care about is minute ventilation. And again, minute ventilation comes in two categories. There's the dead space ventilation, and then there's the alveolar ventilation. Again, this is important when considering pathological lung disease, where you can have an increase in dead space ventilation. So we've talked about the difference between total pulmonary ventilation and alveolar ventilation, and now let's go on to diagram the components of the alveolar capillary unit and describe how these components can affect gas exchange. Back to this illustrated version of the respiratory tract, and let's focus back in on the alveolar capillary unit. Let's go into a little more detail. Now I want you to pause the tape and identify the picture that best represents what occurs during exhalation. During exhalation, what occurs is D, and let's look at that in more detail. You can see in the respiratory tract that you have exhaling CO2 going out to the atmosphere. You have blue blood that is deoxygenated but filled with CO2 traveling to the pulmonary capillaries and alveoli. This is from the pulmonary artery. There, oxygen and carbon dioxide are exchanged, and the red blood travels back to the heart via the pulmonary vein. Let's do another thought exercise. I have listed 6 structures that are part of the alveolar capillary unit, and I want you to arrange them in proper sequence for the diffusion of carbon dioxide from a capillary all the way to an alveolus. There are 2 points to get across using this thought experiment. One is you need to know how to identify structures on an electron microscopy of an alveoli. You'll see it everywhere in the boards. The other one is I do want you to understand the path of carbon dioxide from the capillary out to the alveoli. And this is important because anywhere along the path can be a problem that will affect ventilation. So pause the tape, think about it, and we'll go on. So carbon dioxide travels from the capillary lumen into the alveoli. The first place it must go through is the blood plasma. After the blood plasma, it enters the cytoplasm of the endothelial cell. And once through the endothelial cell, it goes through a fused portion of the basal lamina of both the alveolar cell and the endothelial cells. This is an important part because this can swell during pulmonary edema. Once through the basal lamina, it enters the cytoplasm of what I like to call the type 1 alveolar cell, but you can also refer to it as the type 1 pneumocyte. And then once through the alveolar cell, it goes through the surfactant and finally into the alveolus. Now we've just talked about carbon dioxide's transfer from the capillary all the way into the alveoli, but it's just as important to understand how oxygen goes from the alveoli into the capillary. It's the exact same structures, just in reverse. Now let's focus on an even closer view of the electron microscopy of an alveoli. This shows the exact same structures, with blue indicating the pathway of carbon dioxide and red indicating the pathway of oxygen. P stands for the plasma of the capillary. Then En stands for the cytoplasm of the endothelial cell. B stands for the basal lamina. EP stands for the epithelial cells of the alveolar cell. And A stands for the air in the alveoli. Again, you may see one of these two structures and they will ask you to identify the parts of the alveolar unit. Now let's go to a case. In pulmonary edema, the basal lamina increases due to buildup of fluid. How does this most likely affect the rate of gas diffusion? Pause, think about it, answer it to yourself. The rate of gas diffusion is related to Fick's law of diffusion, yet another physics concept that we need to remember, or, for that matter, relearn. Rate of gas diffusion is proportional to the area times the change in pressure over the thickness. Let's go back to that electron microscopy view of the alveoli. Now, in pulmonary edema, the basal lamina becomes swollen, and this increases the thickness, therefore decreasing the rate of gas diffusion. Let's go to another case example. In emphysema, the walls of the alveoli weaken and eventually rupture, creating one larger airspace instead of many small ones. How does this most likely affect the rate of gas diffusion? Pause, think about it, and we'll get back. Again, we'll go to Fick's law of diffusion. The rate of gas diffusion is proportional to the area times the change in pressure over thickness. Now let's look at a histological view of a normal set of alveoli. You can see there are tons of small alveoli, and the more alveoli you have, the more area you have. Therefore, the more gas diffusion. In fact, that's an important concept, that as you go from one trachea to two bronchus, to multiple bronchioles, to multiple alveoli, you have a huge increase, almost exponential increase in the area for where gas exchange can occur. But then when you have emphysema, which looks like this, you suddenly have very reduced areas of gas exchange, and it would therefore decrease gas diffusion. So, just to summarize, under the learning objectives, we described the difference between total pulmonary ventilation and alveolar ventilation, and why that's important. We've described the components of the alveolar capillary unit, and hopefully you have a better understanding of how it affects gas exchange. Now let's explain the role of surfactant and describe the normal alveolar refreshment. There are two additional features on this electron microscopy that I want you to notice. One is the alveolar macrophage, which is part of the innate pulmonary defense mechanism. The other one is the type 2 alveolar cell. The type 2 alveolar cell, or the type 2 pneumocyte, is where surfactant is made. Now, what is surfactant? Surfactant, again, is produced by the type 2 alveolar cells, or the type 2 pneumocytes. And its primary thing is that it reduces surface tension. We're going to talk a lot about this when we talk about neonatal respiratory distress syndrome. But it's important to know that reducing surface tension does three things. It improves pulmonary compliance, it prevents atelectasis, and it recruits collapsed alveoli. The other thing that surfactant does is it's part of the innate immunity of the pulmonary system. Again, take a look at that electron microscopy. You might be asked to identify what type of cell that is, and that's a type 2 pneumocyte, or a type 2 alveolar cell. And those are lamina bodies, that's what LB stands for, where the surfactant is held until it's secreted and refreshed into the alveoli.